Um, Thank you guys all for being interested in proteomics. This is really exciting to be able to be at a place where, as you're going to learn, it's the biology and the technology, the black boxes, the mass spectrometers, and then I think as important the processing, the software has all come together so that we can do some really cool science. I want to take a moment to kind of just give you a visual of where we are. These were the attendees that are here from all the different places. I put little stars there, and again, forgive me if your star is not exactly in the city that you're from, but I think I got the states right. So, and then we also had a number of people that had applied from all over. And you can see, that's why I was kind of joking with the Kentucky people. There is quite a group of from Kentucky. There's also a group from Idaho. A lot of interest in things, and I have to give a shout out to the lady from Alaska that made the trip down here. That's quite a trek. What is that? How many different planes did you have to get on? It's about three. Okay. Yeah, she had to leave Monday to get here, but that's okay. Last Monday to get here, but you can see so the blue states here, and actually we even had a couple people interested. Those stars up there are not like stars in the sky. There were a couple people from Calgary that I'm not sure if they're online or not, they were interested as well about coming. So there's a lot of interest in proteomics from a lot of different places. The blue states there's nothing political about that. Those are the idea states, and you can see that we primarily advertise to those of you in the idea states, because you understand the pain of trying to get work done in a state where you're underrepresented, I guess is the word, by NIH. We don't get as much money as some of these other guys, but we're okay. We let some people come in from some of the non-idea states as well, because we're glad to all work together to be able to do proteomics and to collaborate. This is one of the best things about proteomics, I think, is it's a place where we sort of come to the end of, you can do a lot of science. You can be a scientist and work a lot of years and basically rely on yourself and your own experiments and your own knowledge. And proteomics is one of those things where you just can't do it all. You have to work with other people. You have to collaborate. And it's really exciting, I think, to be able to be in that kind of an area where you can be an expert in your area, but you have to be able to learn to communicate with the people on either side of you. So that's going to be a common theme as we go on here. So I'm going to start off with an overview here. I think, unless there's something I'm missing, and please interrupt me if I'm not, I think we can just go ahead and roll on. Are we good? Okay. Let's roll on. So we're right here, and we got coffee over here. We've done some introductions here as well. We're a couple of minutes ahead of time, but that's okay, because I've got, I taught for 20 years as a professor, so I was paid to speak, and like every other lecture, I have 484 slides to cover in 30 minutes. Because they're all important. But we're going to try to stick to this schedule somewhat, and for those of you online, we will put a little timer up, so when we do take breaks, you can take a break as well and know when we're coming back. And I also wanted to remind you guys that we are recording this, and we will go through and I will edit it into chunks and put this up on YouTube sometime in the next, when that gets done. Also want to point out that we're going to have a presentation. I wanted to point out, Casey, did you introduce yourself?
I did.
Would you introduce yourself? Just, I think that's important.
Hello, everybody. Casey Brunet from Thermo Fisher. Rick and Alan were gracious enough to let me attend. I'm fairly new to Thermo Fisher and proteomics and omics in general, and this is going to be great for me to familiarize myself a little bit. Please, and dinner last night and dinner tonight, please, everyone join us. That's on Thermo Fisher, and we'll have a presentation from 12 to 1. Please, so you'll grab some food, grab some lunch, come sit down and join us for that presentation. Please enjoy some donuts, and thank you all for coming.
Yeah. Perfect. Thank you. So this is kind of the schedule for today, and I just want to start off with they're so I love working with these guys. So Mike's like, what did you say? Oh, I work with these guys or something like that. He's actually the co-PI of the resource along with Alan there. And the three aims that we have that I think made us successful is one is the service part. That's the part where you get your proteomics done. But the other two, which are near and dear to my heart because I'm all about outreach and education, and that's what they are, is providing a voucher program where you can get your toe in the water and for free. You have internships. We have workshops. And some web resources that are coming on, and all of that together helps sort of create some synergy where this becomes more than just something where we just do this job. Instead, we're collaborating, we're educating, we are sharpening each other as we go forward. And it takes a team. So there are some people that are in this room here that have been introduced. There's some people that are still downstairs prepping your samples or other people's samples. And there's some people behind the scene. Heather Douglas, who she hates this kind of stuff, but she's probably sitting out in the foyer there. She's the one that, got you guys all here logistically. And Sonnet, they work behind the scenes. We've got a bunch of bioinformaticians that also work on the back end of the projects, as well as other scientists that work on the front end. And then we're going to get Sam up here. He's actually on a Zoom call with somebody right now, but he's the front door of our operation. Anybody that does proteomics communicates with Sam, and he does a great job of doing that. But it takes a team to do this with a lot of different skills there. So we're right here at on the UAMS campus in the Cancer Institute. So there's a lot of interaction with cancer scientists here, and we primarily do on this campus discovery proteomics, okay? And we're going to talk about what that is. And also, Mike and his lab over in Oklahoma, they do the targeting proteomics. And so he's going to spend some time, I think tomorrow, giving us some information about that. And there's a picture of his group there. And I just threw this one slide together just to give you an idea, but I took this little graphic off the internet like everybody else does, but as long as I put that little thing down there, it's okay. And so for targeting, he uses things where you can validate. That's really what it is. It can validate up to 20 proteins in a biological sample. So we've gotten to the point where you can use global proteomics to find candidates, and then you can do targeting proteomics to do the validation of those candidates. I'm not against Western blots, but I'm just telling you that this is what they do over there, and it's really exciting to be able to get absolute quantification, is what he's going to talk about, and how he runs those experiments. So what does the experimental workflow look like? Maybe some of you guys are not exactly sure how all this fits in when you say, okay, I'm going to do proteomics and I'm going to work with these guys out there at the National Resource. So if you're a principal investigator, you have your own lab, you do your own experiments, some with cell culture, some with animals, some with populations, all of that. And you have your own science, your own biology, your own physiology. And it's really interesting to hear little, 3 to 5 minute sound bites about what you're working on and all of that. But you do your own experiments after communicating with us, and then these samples get sent to us, and everything in that blue box, we have all three of those parts covered. And so a lot of people think, well, if I have to do proteomics, then I have to do my own digestion. I have to, get those samples ready for you. Because our core facilities, where you have to have the samples completely ready and you literally hand them to somebody, and then they turn around and put them on the machine or the instrument or whatever. That's not the way it works. We actually have our own sample preparation. In fact, Robert is up here for the next couple of days. He preps a lot of samples with Lisa down there. And then what we call proteomics, that would be the mass spectrometry that we do, protein mass spectrometry. And we don't actually do proteins, we actually do peptides. And we're going to talk about that we break everything down into peptides before we analyze that. And then we've gone from biological information, and now we have digital information And then the bioinformatics has the joy of putting all that back together into whatever experiment that you have. You know, what are the controls? What are the treatments? And then and seeing what's significant there. And then they also include these differential expression is usually the data that's sent back, and there's some visualization tools. I'm going to keep covering and recovering this kind of information several times of this morning to see if we can get it to sink in a little bit, because I know some of these words are kind of brand new for you, and you're kind of like, okay, he said something back there, and I don't really understand what that means. So just hang in there. So we'll just take a step back. How do we help researchers? So, for instance, I just have a couple of examples here. We had Aaron Story, who worked with us and has now moved on to industry. He is in the middle of creating, finding biomarkers, and so we can collaborate with clinical samples. There's a kidney repository in town, and so they have lots of kidney samples, lots of kidney disease that they're interested in, and he sort of got lured away to go be their director of mass spectrometry over there. But we work with clinical samples all the time, biological fluids, and that kind of thing. We also, this is actually an old paper. This ProteoViz is a tool that Stephanie and Aaron and these guys made here in-house to be able to visualize your data. We have several of these, and we're going to talk about ProteoDA, I believe, tomorrow. So that's another tool that you'll be able to actually use once you get your data.
I'm going to stop you right there. ProteoViz has kind of been replaced by ProteoDA, so I wouldn't actually utilize that one right now. So,
I'm going to make a note to update this slide. Awesome. And then final, you know, the little box over there on the side, one of the things that we do, and I heard several people mention something like this, is we offer letters of support. Some of you guys are writing different grants, training, fellowships, those sorts of things, and you'd like to be able to say, hey, if I could get this money to do this work, I know these guys, this group that will help me with that. And Sam writes lots of letters of support for people. And then we have collaborative efforts that have spanned years. It's like family sometimes with some of these things. And so we take all of those relationships seriously and do the best we can. Also, professional advice. We have lots of consults, a lot of time where people will call and we'll have a Zoom, and then we may not hear back from them for 3 months, and then they hear from them again, and then, you know, and all of that. So this paper here is just one of those examples. This researcher out at North Carolina, Greg Wong, he's doing really great things, and he's included proteomics as part of a whole bunch of different things that he's doing. And it's just, you know, because he made it into Nature, we like to tell you about that. So here's how the whole flow goes. You guys are all over across the country. How does this work? Do you send the samples first? Absolutely not. Usually it starts with an email, and that gets followed up with a video conference. And we want to talk about your experimental goals, your designs, your sample types, what your group comparisons are. We are not in the business of having you just clean. Well, I've got these samples in the freezer that this other guy had, and we just want to send them to you and just sort of see what's in there. Those kind of experiments don't work very well. We like to have goals set aside up at the front. That makes the best experiments. So then we've gotten to the point where we actually make you send the sample list. So here's the list of samples that we're going to send and what kind of media they're going to be in and the approximate concentrations and all that kind of stuff, so that we can say, yeah, this looks good. Then you send the samples to us, and then everything in blue, this is what we do. We can do lysis and protein quantification, and we do reduction alkylation. This is all part of our standard workflow. We remove the detergents. Why do we have detergents in there? Well, proteins get solubilized with detergents, and so then we have to remove those back out again. We do enzymatic digestion because, again, we're going to analyze peptides, and we're going to talk about these peptides here in a minute. And then sometimes we do isotopic labeling. Sometimes we do enrichment, depending upon the workflow that you're doing. But again, the people in sample prep, they cannot resurrect your samples. If you send us samples and there's, you know, a ton of non-ionic surfactant or something in there. A lot of times all we can do is just put them to rest. We just can't, we don't want to get to that point. That's why the beginning communication is really important. And I know I'm dealing with a group of scientists here, and communication is sometimes not our forte. So getting all that up ahead is really great. And will make a successful experiment. So then once we get peptides out of that sample preparation area, then we start talking about looking at analyzing those, doing the data collection. And so we want to be able to separate those peptides out. And so we do chromatography on the front end. We want to be able to identify those peptides. And we do that by mass to charge, and then also by the fingerprint that we're going to talk about, or the fragmentation that we get. We want to be able to quantitate. So we're way past the point where we just want to know what the protein is. We want to know how much, right? We want to know the quantity, quantification. And so we take all that information, and then we do a database search, and bioinformatics can put all that back together in a final report. And believe it or not always, but we're pretty good at getting that all done in about 4 to 6 weeks, okay? And that whole cycle there is what we call our workflow. And there's going to be information, those things in the red boxes, that's communication back and forth with the PIs. And so there's a lot of communication that goes on here. So what type of proteomics do we do? Well, in the jargon is we call it bottom up, right? So we don't look at full proteins. Proteins are really sticky. They don't analyze well. So what we do is we use trypsin, which is a proteolytic enzyme that will digest that, and then those proteins, then we get, those proteins are first reduced and alkylated, then they're subjected to trypsin to make peptides. And we're really looking for all of these different types of workflows that we have. There's 2 basic types of information we get. We get the MS, which is scan, and then we get the MS2 scan here. And then from that information, we do a database search to get peptides, and then from the peptides, we have a library that we can go back to proteins here. So just so you know, if you're talking about an MS signal, that is a scan, and we're going to talk about this more again, but it's a scan where each signal then represents a precursor or a peptide. So each one of those little lines there is a different peptide or precursor. And the word precursor and peptide are kind of synonymous. They're a little bit different in nuance, but you can think about them as the same. And so then what these mass spectrometers can do is they can not only just say, well, here is, by scanning across that mass to charge range and figuring out all of the things that have signal there, they can then isolate, depending on the workflow, each one individually, and be able to take and hit energy to make a fingerprint here. So then each signal down here in the MS2 represents a different fragment of a particular peptide. And so the old school way would be that if you were a peptide mass spectrometrist, is you would look at these MS2 signals like this, you would have a list of masses for your amino acids, and you would go and say, well, the difference between these 2 masses happen to be alanine. And you're like, okay, I've got an alanine in there. And then you'd go and do the next one. It's like a little puzzle you could put together. Nope. Okay, thank you. So we start off with biological information over there with the proteins, and we end up with digital biological information over here. And that's the beauty of this, is we're trying to get, I mean, you can look at your sample with your eyes, but you really can't see what the proteins are in there. And we're trying to be able to see what that is in a digital way, in the best representation possible. So I'm going to go through here and just talk about a couple of the main things. First of all, we always do reduction and alkylation. Reduction means that there are some cross-linking that goes on in proteins between the sulfurs on the cysteine residues, and we want to break those. And to keep those from coming back together again, we then add some iodocetamide, which will then alkylate them. That's important for a mass spectrometrist because you have now added mass onto certain residues, and you have to account for that in the database search. So if you're wondering, hey, why is it that I have this extra mass of 57 or whatever, that's what it comes from. And this is, Let's see if I can figure out how to get that out of, okay, there we go. So one of the things that we use in proteomics that's really key is, and you may not know this, is that we don't actually, here anyway, we don't do things, what do you call that? Like if you were using peaks or something and you're sort of building it up from nothing, and you're basically saying, I'm going to create all of the peptides themselves and then try to put them all back together as proteins. We sort of cheat a little bit and we start with a library. So we basically go back to UniProt or something like that, that is a repository that says from this genetic information, from the genome for whatever species that you're talking about, there's only a certain finite amount of protein that can be expressed. And so I always like to think about like this. It's like, really, there's only about 81,000 different proteins that you have to make and very specific things and modify correctly. And then you could have a human cell, all the human cells, and you could build your own human that way. But there's only about 20,000 genes there. And so I'm going to let those people that are a lot better at genomics and that explain why there's 20,000 genes, but there's 81,000 proteins. But what that means is that each protein has something that looks like this. This is what's important to us. It's not genomic or genetic information, it's proteomic information. So for instance, this is just one of the entries out of the human FASTA file, and this is for the insulin-like growth factor, and that list of alphabets there, each one of those represents a different amino acid, and there are a whole bunch of them, and this is just the primary sequence of that. Now, where is it cross-linked? Where are the other things that are attached onto it? We don't know, but we do know the primary amino acid sequence of this particular protein. So let's go back to the workflow again, all right? So you bring us protein in some form. That could be in the form of a gel. People still run gels. We run gels all the time. Biochemists like gels, running gels, because they can see the little bands and they know that that's where their protein is. And they have a ladder over there that tells us what the molecular weight is so that they can do that. Or we do what's called a chloroform methanol extraction, and that's where we can take a cell lysate and basically sandwich between two immiscible layers the protein pellet, and then we can wash it and be able to take that protein pellet and be able to digest it. Or we have these little tiny, those are little filters that are called FASP filters, and people in sample prep love FASP, right? Right, Robert? So funny. FASP protocol takes a lot of just sitting in front of a centrifuge watching that thing spin. So basically it's a little filter that you collect the protein on, and then you wash it, and then you can digest it right on there. It doesn't matter what form that is. That can be, we can do all of that, but we add trypsin, and once we add trypsin, all of that protein, no matter how it was originally collected, all becomes peptides. And those peptides are all charged. And those charges then allow us to be able to route them through the mass spectrometer. But another thing I want to point out is that the sequence of those different peptides are going to include different types of residues. Some of those amino acids are polar, some of them are nonpolar, some of them are charged, and so that means they're going to separate and reverse phase chromatography based upon that polarity. Some are more hydrophobic, some are more hydrophilic. So we get some separation, then we put them into, when they come off of the column, into the mass spectrometer. And it's really kind of, I think one of the most amazing things is that the liquid chromatography and the mass spectrometer, they're not actually attached. There's about a centimeter gap where you're just basically spraying out these peptides, and then there's a connection by a voltage connection between the column and the mass spectrometer that then brings those ions into the mass spectrometer, dries off all that solvent. Now you have all of these gas phase ions that get routed through the mass spectrometer in this amazing way. And that's where all the black box stuff comes. But again, we only get really two things that we're looking for. We get the MS signal, which would be a scan, and that tells us the mass to charge ratio of the peptides, and then we would get the MS2 so that we can do our database searching, right? So let's dive just a little bit deeper. So when you think of trypsin, you think of like a great big pair of scissors that that selectively cuts or cleaves at the K or the R. So that's going to be the lysine or the arginine residues, and that's going to be on the C terminal side. And so if you take your amino acid sequence for our insulin-like growth factor here, and I've color-coded it in red and blue, that would be all the potential places where trypsin is going to go and cleave that guy up. Now, what happens in reality is that your protein sometimes is folded up, or trypsin itself is I don't know what its molecular weight is, but it's got some size to it, and so there's some steric hindrance, and it can't get into all the places. And then there's some other rules that some mass spectrometrists will tell you about where it can cleave, or biochemists will tell you that it doesn't cleave with a proline nearby or something like that. But those are all the possible peptides that we can make. So a lot of people are really, you know what I forgot to do? I forgot to share the slides with you. I will at the first break, I promise. I'll share slides. But you guys are doing great if you're taking notes. You get a little sad if you don't see all the peptides, right? I know there's proteins in there, but I don't see all the peptides. Why not? Well, first of all, because some of them don't fly well, right? There's a range of the size of peptides that will be able to get into the mass spectrometer and come off in the range in which we have the instrument turned on. And so some of them are just too small. I believe that if you have a 4 amino acid peptide, we're never going to see that. It's too small, okay? And some of them are too big. They're too big, and so then the question is, well, how many unique peptides do I need to have for my protein before I can say I've got that protein? And I think the answer out in the now, if I look over at Mike, how many peptides do you need before you need 2? Rick's a little bit, he says 1 unique. And when I say unique, so now we can get in the thing of, we're talking about proteins, or we're talking about protein groups. When we say unique peptide, we're saying that sequence is only found in that particular protein in the entire in the entire library. And so there are some questions there, and you as the investigator can decide how far out you want to go out on that limb and stake your scientific reputation on a single peptide indicating a certain protein that you are now saying this whole pathway is whatever. So, but the other thing is, not all the peptides will be ionized, not all of them will be detected, and there'll be gaps. So for instance, if we do collision-induced dissociation, which is how we fragment up those peptides. So for this, when the peptides go through, we hit them with a certain amount of energy, and it breaks apart into fragments. So this spectra that you're looking at over here is, I don't know if this works or not. Oh, that's because I'm pushing the wrong button. This is the MS2 spectrum, and you can see that now it's color coded and it has these letter things up here. So what I've done is I've found a single peptide from this human insulin-like growth factor, and we're looking at this at the MS2 spectra for it. So this is the information that's coming off of the mass spectrometer. And then after database searching, it has determined, because this spectra matched really well experimentally, and the observed spectra matched really well with this sequence right here. And that sequence happens to be from this protein. And so this box of information over here is telling you, these are the calculated, for this particular sequence, these are all the calculated masses, mass to charge that we would see over here. And the ones that match up within whatever error tolerance that we have, which is usually like 3 or 5 parts per million, or no, not for MS2. What is it for MS2? Yeah, maybe 10 ppm. that matches, it turns them either blue, which is showing you all the Y ions here, or red, which is showing you all the B ions. Now, all that means, the B ions and the Y ions, is that if you take the full, you might be able to see up here on the top, this is the full sequence for that particular peptide. And what it's showing you is that wherever when you hit it with energy, I don't know if we just got, you can call it whatever you want, but I think it's pretty fortunate for us that when you hit it with energy, these things tend to break right along the peptide backbone. So each amino acid, when it breaks off right in the middle. So the mass difference between these guys are the mass of the amino acids that you see up here, and you can calculate from the mass difference all of that. And computers are great at this. Now, the Y ions just mean that the charge happens to be counting from the C terminal, and the B ions, I think it's from the N terminal. But a couple of things I want you to point out is that sometimes when you talk to someone and they look at a spectra, they go, well, that's not a very good spectra. And you're like, well, why is that? And they're like, well, there's not very many Y ions up in this range here. Can anybody tell me why you might want to find these guys over here instead of hang your hat on these B ions over here? You didn't know there was going to be a test here. Yeah. So, my training is in analytical chemistry. So when I first look at this, the first thing I notice is the noise. And I'm like, wow, there's an awful lot of noise at this end over here. Not as much noise over here. The other thing I notice is the size of the peaks. Yes, ma'am. Oh, that's a great idea. Sorry about that. Yeah. Isn't there a little thing down here where I can get? I can't see it though. Is it that one? Yeah. Laser point. Okay. Sorry about that. Yeah. So I would say that so over here, the size of the Y ions tend to be taller. They're more intense. The B ions tend to be smaller. You can see they're down there lost in the noise somewhat. So then the question is, okay, so we said, how many peptides do you need to be able to declare that you have a protein, right? How many unique peptides? So now the question is, how many fragments do you need to be able to find before you can say that's peptide, that's a legit peptide? And that's a question as well, because as the biologist trying to explain all the way back at the organism level, and some reviewer wants to take you all the way down into the weeds there, right? And so these things are kind of standardized. So what do we say? How many fragments do you think we need here? Rick, what would you say? He knows I don't know the answer, and that's why he's laughing.
It depends, right? So the best answer in the world. You want a string of fragment ions in a row to help you feel better about the mass differences leading to a unique stretch of sequence. And so, at least 3 ions in a row that's going to give you 2 amino acids. So, 4 ions in a row, but sometimes you don't always see that. So that's preferential, but not always.
Can I just throw out a question?
Sure.
Please.
Cool. So larger proteins should have more fragments. And so do you take that into account? Like one peptide from a large protein is less convincing than one peptide from a small protein.
Correct. Yeah. And so it was a trick answer a while ago when Dennis asked me, how many peptides does it take? And 2 is a good safe answer. And I say 1, you can have 1 peptide and you can be completely confident in a protein, but there has to be a reason why that you only see 1 peptide. Mm-hmm. And it may be that protein only has 2 possible tryptic fragments that are in the mass range that you're looking at. And if you see 1 of them, you're seeing half of the observable peptides. Mm-hmm. And, just because you can be confident on one peptide doesn't mean that every time you see one peptide, you're confident that that protein is correct.
Okay.
And fragment ions are like that too. And so no one does this anymore. So, I mean, you know, I'm old enough to know that when you did mass spectrometry on peptides, the way you got your answer was with a calculator or a good memory and a ruler. So you're measuring mass between the sticks and the spectra. But now it's all done with some type of correlation mapping to a known database. And so there are a number of different algorithms that we use to search this. But it kind of comes down to when your biologist asks you, hey, this looks really interesting. We want to knock this gene out and spend 6 months in the lab studying this. That's when I want to see the spectra, right? I want to be confident. And I've always said there's three kind of spectra. There's ones that you're convinced of. There's ones that you could convince your mother of, and then there's one that you can convince a reviewer of, right? And so the truth lies somewhere between them, right?
Yeah, that's right. Good deal. All right. Thank you guys for participating in that. So the other thing I want to point out here is in where the primary sequence is up there, highlighted in gold. Those are the peptides that were found. So we didn't have there's not 100% coverage, even though this protein definitely was there. And it's because of all those reasons that we just talked about, that it's some of these are too small, some of them are not good flyers. And because Mike's made a living off of targeting, he can look at, that's what he does. He would look at those peptides individually and figure out which one of those guys are most likely to be what he calls good flyers. They're peptides that we're probably we should see. And so then he would then target those to be able to validate a particular protein there. So there's that sequence that we're talking about, is that little guy right there. Okay, so let's talk about post-translational modifications just a little bit, because people are really interested in these. And at this point, there are different groups doing different types of modifications. Of course, you know a lot more biology than I do, but this chart here shows that there's lots of different things that you can hang off of different parts of the protein. So I will say this, because sometimes when people will ask and they say, well, can you do glycoproteins? And I'll say, we don't. And I'll say, oh, so then the question is, so you can't see a protein, that has some sort of sugar hanging off it? And I said, well, actually, we can see peptides from that protein, but we're not going to be able to see that sugar that's hanging off of that peptide. And for a lot of these different modifications, like I mentioned before, where we purposely modify the cysteine by acetylating it, all we have to do in our database search is just tell it, hey, every time you see the cysteine, expect there to be most of the time an extra 57 on there. We can do the same thing with some of these guys, like methylation. There may be some methylated arginine. And you can say, well, let's just do a variable search so that, and that's an easy one. But what we have to be careful of, and I'll let Stephanie talk more to this if she wants to when she talks about the bioinformatics, is that people will say, okay, great, I want to look at all the modifications. The problem is when you go looking into that space, it just makes it all really hard, because now every single possible combination could fulfill something. And so that makes it a lot more difficult. What we do here in the National Resource is we do routinely look at phosphorylation. That workflow is pretty standard here. We do an enrichment to try to pull out the phosphorylated peptides, which is not many, like maybe 1% of all the peptides. So we have to start with a lot more material to be able to do phosphorylation. And there are other places in the country will do some of these other things. So let's talk about the word discovery, because I want to make sure that you guys have in the right frame of mind about what type of experiments, what the goal of the experiment is. Joe here from Rhode Island, who's been here the last couple of days, and we were having some different conversations, kind of prepping me for this, and kind of reminded me that, when you're doing proteomics, you're really looking for candidates, right? That's the goal. This is not a definitive, I'm going to explain all my biology with this one single experiment. So discovery proteomics are designed to quantify and identify the greatest number of peptides and proteins in a biological sample. And it's given us sort of like if you're a detective, it's giving you leads to go look down and say, well, that's really interesting. I'm not really, I didn't know that protein would be overexpressed or underexpressed for whatever biological change that I did. I'm going to go see if that's real. I'm going to go see if that has something to do with this biological story that I'm trying to tell here. And sometimes you don't find what you want in a discovery experiment. And so we can always say, if you want to go deeper, and that means find more candidates, then you can do fractionation and spend more time, more instrument time, okay? But more times than not, unless you know what you're looking for, all that's going to do is just give you usually our sort of our normal standard workflow is going to give you lots of information to go look at. And so unless there's something that is really low level, not normally found in your standard workflow, there's no reason to do that unless you really know what you're looking for to go deeper. So let me just talk about this, some of the challenges we have. And I was trying to put together something with just cell lysis, but we're going to talk about the plasma proteome here for a little bit. It may be hard to read, but this is a list of some of the different proteins that are found in plasma here. And over here on the left-hand side, this is a log of the concentration. So there are 10 or more orders of magnitude in the protein concentration difference for the plasma proteome. And that just means that the real challenge here is the dynamic range, okay? All the ones that everybody's interested in tend to be down here at the bottom, right? Not very concentrated. But what we have, it's like, and it's hard to see because it's cloudy today, but like if you were to look up there in the sky, and I say, how many stars do you see up there? Right now, you'd say, well, I only see one. It's the sun. And that's basically the problem. If you don't remove some of these really highly abundant guys, then that's all you're going to see. So because of that, we with plasma and some of this, we want to deplete that out. And so that's one of the challenges, is that if you have something that you're looking for is really low level, the more abundant things are going to be distractors, because the mass spectrometer is going to be looking at those things and not looking at the low level things that they're coming out here. Okay, we're hitting a bunch of different little topics here, but the next one I want to talk about is and this is my opinion here, but what is my opinion of a good experiment? A good experiment, and of course, this is all from things that I've heard over the last couple of years here, is good experiments have simple or few comparisons, okay? We tend to get, people sometimes want to just put everything into one experiment and try to do, 14 different contrasts, and that tends to really dilute down and make it really difficult to really answer. So when you're doing your experiment, try to ask the most obvious question first. The other thing is the statistical power. We like to say that 5 is the new 3, okay? Having 5 replicates, or having enough replicates that we can measure the variability within the group. So for instance, a simple experiment, a proteome experiment would be a 5x5, where you've got 5 controls and 5 treatments. And that may be, for some of you that do cell culture, you're like, that's overkill, right? 3 is good enough. Okay, that's fine for you. But remember, we need to have enough replicates to be able to measure the in-group variability, so that when we are trying to find the difference between the groups, we can be confident of that. There are two main workflows that we are that we're going to talk that we do here that I'm going to just touch on a little bit. The first one is it's a DDA experiment where very much like what you just saw before, we take an MS1 and we do an MS2 where of the fragmentation pattern here, but we also include a tag label on the end. It's called a TMT experiment. And basically, I'll talk about this in a couple of different ways, but you start off with taking your sample and adding covalently bonding to each sample on the end of every peptide, a different molecular tag that will then stay with the peptide all the way until it's being routed through the mass spectrometer. And then we will be able to do our quantitation based upon the measurement or the intensity of those different tags. And the advantage of this is it's quantitative, it's applicable to a lot of different experiments, and we call it multiplexing. We basically get a lot more bang for the buck. You take 10 different samples and you essentially are able to combine them all together. So the concentration of each peptide is greatly increased. And so then we can run fractionation over that. And we usually use MaxQuant to do a database search. And then, am I right? We still do the Proteo DA on the back end so that we do the normalization and QC and the differential analysis. So disadvantage of doing TMT, because you're like, oh, this sounds great. Why doesn't everybody do TMT? Well, it's kind of expensive because those molecular tags are expensive. And then also we're limited on the number of tags that we can add. So you can't do, it's hard to do an N of 40 or so, you really 20 or less. So my children did not make this, I did. This is this is what I think a TMT workflow looks like. So these little squiggly lines here represent the peptides. So you've got, and I didn't draw up like 10 samples or even 6 samples. I only drew 2, but you take your peptides, you go through this labeling where you put a tag on there, and it's really, really slick. If you look into the TMT thing, it's really cool because they have really designed this in a very elegant way, and it works quite well. But you get these different tags on all the different peptides, and you're able to combine them together, and they make different kits. So you can do a 6-plex or a 10-plex. They've even made an 11th label. And then they even have what's called a TMT Pro, which are 18 labels. So that means you can do an experiment with 18 different samples, and there's a molecular tag that would go on to 18 different things. So what we normally do is combine all that together. We do offline fractionation. And what I'm showing you down here below is kind of what offline fractionation looks like. So when you put something into when you put a sample into the mass spectrometer, the sample is gone, right? It destroys the sample in there. But when you have an offline fractionator, you basically you separate it out, you look at it by UV, and then you basically take the sample and put it back into different fractions. So you still have it. You're not losing it. And what we're able to do is we're able to take those fractions and recombine them into 18 different parts, putting some hydrophobic, some hydrophilic, some in the middle, so that even though so that every time we do a run, one of these runs on the mass spectrometer, we're going to have the whole gradient is going to be filled with peptides. And Rick has got this. He would love for you to ask him about this fractionation and how he combines these all together again and does that. But for a TMT experiment, we really get 3 pieces of information. So you got the MS experiment over here, which is still that same. Each one of these mass to charge of each peptides is each one of those peaks represents that. Then we hit it with some energy, and we break it apart into fragments. And so this fragmentation pattern, this MS2, is going to tell me the sequence of the peptide that we just analyzed. But then it hits it with more energy, and it breaks off those tags, and then it gets a signal of the tag. And so here's what all those tags look like. And so the relative intensity of those tags then tells you the quantification for that particular peptide. You want to jump in there, Rick? Is that okay? Okay. All right, so those are the 3 steps that we do for a TMT experiment. The other version, I'm just gonna, I'm not gonna talk about DDA experiments per se. The other one I'm gonna talk about is DIA experiments. This is the kind of the new kid on the block. And all the instruments that have been put out in the last couple of years that are coming out are really geared for these types of experiments. So a data independent acquisition experiment it does not involve a tag, it does not involve a label. It is also quantitative. It is also applicable to lots of different types of biology, but without a tag, it's a little bit cheaper. And it allows us to be able to do lots more samples. Where we're limited in the number of samples for TMT experiments, we can do hundreds, we can do thousands of samples in a single experiment with DIA. And so that involves using a slightly different instrument. Instead of using the Eclipse, we use an Exploris, and it's geared towards that. Instead of using Max Quant, we currently use Spectra to do our database search. And then we still do the back end, the ProteoDA, the QC, the normalization, all that kind of stuff. So you'll get a very similar looking output for that. The disadvantage is we don't usually fractionate, so we don't get that added boost of being able to have all of our samples labeled and put together so that we get elevated peptides for each one. And to say that the database searching is complex is just putting it mildly, okay? Whereas before, when we looked at an MS2 spectra, I could give you guys a scale and we could all figure out what this peptide, the sequence of this peptide is. With DIA, it's really difficult because you're going to have lots of things being fragmented at the same time, and a computer has to then be able to go through and do that analysis for you. So here's kind of what it looks like for the DIA. You still get the same, we still do the MS scan, where we scan across all of those master charge, and each one of those represents a different peptide. But here's the thing, we'll take a window like 12 Daltons or 6 Daltons or 10 Daltons or 2 Daltons, depending on how sensitive and fast your instrument is, or 20 Daltons, whatever. And we'll take a mass range from like 400 to 412. Every peptide that has a master charge of that between 400 and 412 is all going to be fragmented at the same time, and all of those fragment patterns are going to come out. So what you're looking at here in this MS2 window is several, maybe 10, maybe 20 different peptides all being fragmented at the same time. And so this is where sort of the old school mass spectrometrists that could kind of like feel in their gut, yep, I know this peptide because I can look at the spectra and everything. This is where it kind of goes, I can't do that anymore. I need some more tools to do that. And so then we would do those windows from, 400 to 412, 412 to 424, whatever, and we would go all the way up, and then we do another MS scan, and then we'd start it again. And so we would then be able to cover the whole range that we have here. So here's a picture view of that as well. You've got your peptides coming in here into the quadrupole. It is isolating, for instance, in this example, 25 Dalton window. It's fragmenting all of those at the same time. It's getting this really complex looking MS2 intensity. But if you look at it with the retention time as the third dimension here, you can kind of see that those fragments are actually going to kind of line up based upon what peptide they're from. And so now you can start to say, oh, look at these. All the purple guys are fragments from the same peptide. And so a computer then can score that like we do other scoring mechanisms, and we can get some information about what peptides there are. And then the difference is we quantitate on the fragments. Okay, we don't. That's our quantification is we take a couple of those fragments in the area underneath the peak of those. I believe it's 3. Is it 3 fragments? Yeah, that we quantify on. And so DIA is really good for plasma and serum in that a higher number of samples. The protein depth sometimes is not as good as TMT that. Okay, we're going to keep going here. You guys are doing great. Yes, please.
So, I think I get the MS.
Can we get you on a mic? I'm so sorry.
So I think I seem to get the MS2 scan and that it's a fraction and you have a fairly limited range of things that you're looking at. I guess I'm taken aback by the MS1 scan, and that seems like a much broader range. And if there's 80,000 possible proteins in a sample, that seems like a relatively small number of peaks for the, for the range of mass to charge ratios that I would expect.
Okay, okay. I would say for like on a normal, the number of precursors that we might get off of using our Explorus for an hour DIA run for a lysate might be on the order of 60,000 precursors. Because that FASTA file said there's 80,000 possible proteins in the human proteome, but in any particular cell, there's only going to be maybe 10,000 cell proteins that are actually expressed to make that cell function, and out of there, we are able to measure maybe 5,000, 6,000, depending upon the sensitivity of our instrument, and it's getting better. Some people say now we can do 10,000, all in one scan or whatever. Does that answer your question? Or what are you getting at
Yes,
here?
well, your answer answered my question, but the data makes me kind of wonder about that because I'm just not seeing 5,000 peaks in that, figure that you put up there.
And
Yeah, like that. Okay, so the MS scan, I'm not seeing 5,000 peaks.
okay,
And there's some that are like way,
Yeah. So that's one MS scan from a tenth of a second from an hour-long chromatographic run.
yeah.
Yeah.
well, that's a different kettle of fish.
Yeah.
Okay, all right.
Yeah, that's a snapshot of a fraction of a second of an hour-long run. Yeah.
I had a quick question too. Yeah, I've been amazed at what you guys have produced from our data for proximity labeling and worm. It's sort of a miracle. But what I was curious about is when we did the multiplexing, I just assumed they were labeled, and then Stephanie told me that it wasn't. How do you know? Are you running the sample sequentially? How do you know which peptide came from which sample? Is it because you're not running them all at the same time?
Are you talking about for a TMT experiment?
No,
Or
I'm talking
the
about for the DIA.
Okay,
Does
so
that make sense?
Yeah, so for DIA, we're not mixing the samples together. Yeah, each sample is run individually. In fact, for DIA, it's really important that we actually run them in some sort of random order that we keep track of. Yeah. For carryover and, other things like that. Perfect. Okay, we got just a few more slides, and I'm really hoping that in 10 minutes we'll be able to take a break, because I know the fire hydrant has been wide open here for a little bit. But one of the questions we want to go back to is, now that we've talked about some of the more about data collection and these different types of workflows and stuff, how much sample do you start with? Like, all of that's great, and the number of fragments that I need and all that. I just want to know, like, how many plates do I need to grow up of my cells to be able to do a proteomic experiment? Well, we usually tell people somewhere in the millions of cells, and for fresh frozen tissue samples, I think 100 milligrams would be way overkill. I'm going to show you a picture at the end that basically the amount of stuff that needs to be in the bottom of a tube, and say if you can hit that would be great. Because really, Robert, one of his biggest challenges is when you guys shove half a rat brain in a conical tube and you send it to us. That's a full day's work to make a lysate out of that. And the proteomics is not going to be very good. Laser capture. No one would do that. Laser capture and by the way, every example that I used today happened. It's all real and been done. So we can also do FFPE, and Rick is kind of shocked, and one of these days it's going to come true, and then you're really going to be in trouble. But he's like, how many tens of thousands, hundreds of thousands of slide samples are in freezers and all over the country that we can go back and break those cross links and get rid of the parafilm and be able to look at all those slides that people have, and we do that sometimes. And then serum or plasma, we need about 10 or 25 microliters would be perfect. We've talked a little bit about instrumentation here. We on site we have 2 fusion class Orbitraps, 2 Eclipse class Orbitraps, and 2 Exploris. And and people a couple of years ago thought, do we really need 2 of everything? I mean, isn't that a little bit overkill? How many of you guys have a mass spectrometer at your site where you are now? Okay. Is that thing running 100% of the time? Anyone? Okay, that's what I thought. So that helps us be able to hit that 4 to 6 week window there by doing that. Just some other light things that I want to tell you about. So pricing structure for us for DIA to do 20 samples is just under $2,000. And that is broken up. The cost that's $24 if you have us do the digestion here per sample. So you may have a graduate student or a postdoc that really wants to do that, and that's great, but a lot of times it just will save you time and headache later if you'll just send us the samples and let us do it. Orbitrap Exploris time is about $45 an hour. And then we have a $500 setup fee for the Spectronaut license and other stuff. TMT analysis for 10 samples, it's right at $2,000. And if you add phospho, so the phospho analysis where we do we will do basically you get 2 for 1. You get those 10 samples. We'll do the full proteome, and then we'll also separate out the phosphopeptides and do that as well. So it's like double that, and the price is double. And Rick is the one that almost exclusively works on that workflow. Some of the bioinformatic deliverables that you're going to get that Stephanie's going to talk through are things like a dendrogram, so you can kind of see where the samples, the different groups of your samples come out, and there's a reason why we have that particular plot, and she'll talk about that. The violin plot kind of shows loading, and it's funny because I worked here for a long time before I realized most of our stuff looks exactly like that violin plot. It looks really good because sample prep does an amazing job of making sure that we have equal amounts of abundance for all your samples to start with. And then your PCA plot, it always gives you a comforting feel when you see all the blue dots sort of gather in one side and all the red dots gather in the other, that says that you've got some sort of, difference there that you can see. They also have these interactive MD and volcano plots that she's going to talk about. Some of this data is from Mike's work. He's a big mouse model guy and does this fish oil diet sort of thing, and he'll show you some more specifics on that. But again, if you look at one of those red dots up here and you say, okay, well, that's the protein that I want to look after. I just wanted to give you guys a little bit of time this morning to see all of the layers of data that's underneath that to get that one little dot that's up there, okay? You don't have to be an expert at that, but you got to know a guy or a gal that can kind of walk you through that again and say, can you remind me how that, because I've got this reviewer and I just need to put this little thing out there about that. The other thing that you get for all of you folks that love spreadsheets is you get this differential expression in a spreadsheet form, and it's got the what do you call it? The annotation over there. It's got your protein IDs. And actually, I think she has it to where all of these IDs over here are going to be links. So you can link it back right to UniProt so that you can look at the UniProt information for that. And then she has log fold changes and p-values and adjusted p-values all built into your into that. So you can easily be able to filter these tables to see all your significant proteins and those sorts of things. We've covered a lot. So the question you might be asking is, where do I start? Well, in the next the rest of this time here, we're going to talk about answer some of these questions, like what are the different workflows? We talked about that just a little bit. We'll spend a little bit more time. And how are proteins and peptides identified? We spend a little bit of time talking about that. And then, okay, for my particular experiment, which workflow should I use? Or how are the samples prepared to create peptides? We spent a little bit of time talking about that. And then what workflow is used for PTM analysis if you have a specific PTM that you're able to enrich for or that you're looking at. We can talk about that. And then what are the challenges and limitations of global proteomics? All of these sorts of questions here. What methods are used for quantification? That's really important. Are we talking about fragments? Are we talking about this MS3 stuff? And then how is the experiment validated statistically? That's fairly important on the back end there. And then and how is the data visualized? And basically, more importantly, it's just where exactly do I start with all of this? And so I love this part right here, but that's the guy right there. So Sam will be up here a little bit later as we get closer to lunch. But that is the guy that is our front door to our resource. And of course, Stephanie's also available as well. But a lot of times, if you have questions, we start with sending an email to ideaproteomic@uams.edu. And you set up an initial consult. And I know that many of you guys have already started that process. You've gone down that road and everything. And that is kind of where you get started with all of this. And he, a lot of times will then schedule a Zoom call and we'll bring Rick in or myself or Stephanie, if there's something that we need to talk about. We have several meetings a week where we talk about projects and bounce ideas off of each other. And so it's a very collaborative sort of thing. Do you guys have any questions? I think we have 2 minutes till break. Yes, ma'am.
So is it common to use HCD for protein fragmentation, high collagen dissociation?
That's a Rick question. ECD, is that common? Do we do that much?
Or CID?
We do mostly CID.
I can answer that right after the break.
Okay.
Okay.
Yes, sir.
So let's say there are two proteins. The one protein is a full parent protein and the other protein is a paralog, which is a truncated form of the parent protein. So can you differentiate the two?
Stephanie?
Only if there's a unique peptide between the two that's mass spec compatible. So if you have that question, you need to we want to look at it beforehand to make sure that there's a sequence difference. Otherwise, it's going to put it as a group and it's going to have shared peptides across the proteins and you can't distinguish the two. So
Okay.
it's going to depend on the protein sequences. But always good to think about beforehand.
I kind of have a follow-up to that. Do you ever run then controls of just recombinant protein? Like if you had the truncated and the full form and you ran those and compared them to samples, would you be
Feel
able
like
to?
we've done that before, but not often. And that might, if you have proteins of specific interest, that's where I would talk to Mike on the targeted side. Because this is really meant for discovery to find everything that's there. But if you already know what you're looking for, then we can just tell it, hey, we want to sequence this and make it a lot more quantitative that way.
Yeah, it would be a theme, and a lot of this is a lot of what you're hearing, not to discourage, hopefully to still encourage questions. A lot of what you're talking about here is a high throughput processing of samples. And so, what then is the next step is for the investigator to look at those data and then start to ask those sorts of questions. Because a lot of times exactly those answers are buried in the data set. And problems are solvable. We just need to be pointed in a particular direction to be aware of that and to understand that. Differentiating between isomers, if that's important, are things that sometimes, could be a little bit lost on us as we just plow through all these identifications that come from the experiment.
Stephanie, is there a comment or a question online?
Yeah, I'm answering questions in the chat
Oh, thank you.
and yeah, so
Perfect. All right.
Just
What-
an extension to what has been recently talked about, like, there are a lot of aberrant proteins. So can we kind of distinguish between the aberrants or, consequence of alternative splicing? Can
Yeah, so
get to-
alternative splicing stuff and isoform, again, it's gonna be the
Okay.
protein sequence. So yeah, we've got to find that unique peptide that's gonna separate those. It's a much harder thing to do than, yeah. But yes, similar. There
Okay.
is a question in the Q&A.
Okay.
What's the minimum Dalton window that can be used in DIA analysis?
What's the minimum what?
Dalton window that can be used in a DIA analysis?
So it's really a question of how fast the instrument is to be able to scan across the range. If you're only looking for specific proteins, you can go down. If there's specific proteins and then it almost becomes a targeted method. We use a 12 Dalton window or Thompson window for our workflows on the 480s because it's a good number of points across the peak. So it takes about 3 seconds for us to scan across the entire mass range, and then our chromatography such that we can get enough points across the peak. The new thermo instrument, the standard method is using a 2 Thompson window, and so that's the smallest that I've seen anyone use. And I guess you conceivably could go down smaller than that, but somewhere between 2 and 50 Thompsons is standard. 10 to 20 is probably most common.
Okay, I know you guys might have more questions, but let's go ahead and take a break. We can answer questions as we come back, and we'll